In this video lecture, we will continue with the remaining commonly used media ingredient such as growth factor, buffer, precursor, inducer, inhibitors and anti-forming agent. So, let's start. First, we will discuss about the growth factors. If we talk about the growth factors, microorganisms are not able to synthesize. They are all growth factors. Microorganisms are not able to synthesize all their growth factors. So, we need to supply some of the growth factor in the fermentation medium. The most commonly required growth factors are the vitamins, amino acids, fatty acid, sterols. These are the most commonly required growth factors. If we talk about the growth factors, many natural carbon and nitrogen source contain all or some of the required growth factors. But sometimes we need to add pure or artificial source of growth factors. Natural medium containing, if we are adding some of the natural media, that itself has a some or all of the growth factors, whereas sometimes we need to add pure or artificial form of growth factors. If in an industry, if we are producing some product in an industry, if there is a requirement of only one vitamin is required, it is more economical to add a pure vitamin, because instead of using the cheaper or the multiple vitamin source, it is economical that we add the pure vitamin if there is a requirement of one vitamin. If we talk about the calcium pantothenate, it is a vitamin B5 that is used for the vinegar production, whereas biotin is the vitamin B7, it is used for the glutamic acid production. Calcium pantothenate, it is a vitamin B5, it is used for the vinegar production, whereas biotin, it is used as a growth factor, that is a vitamin B7, it is used for the glutamic acid production. This is what all about the growth factors. The things that we have to remember about the growth factor is that microorganisms cannot synthesize all the growth factors. So we have to add the growth factor in the fermentation medium. The most commonly required growth factors are the vitamins, amino acids, fatty acids and the sterols. Calcium pantothenate, it is a source of growth factors. It is used for the vinegar productions. Biotin, that is a vitamin B7 that is used for the glutamic acid production. Buffers. The main function of the buffer is to regulate the pH. Buffers are used to regulate the pH of the fermentation medium. pH can be controlled by sometime natural medium or sometime artificially buffering agent we use. Sometime we use the natural media or sometime we go for artificial buffering agent we add. If we are using natural medium, it has a dual function. First, it will act as a nutrient source that will provide the natural media that will provide the nutrient source, and it has a greater buffering capacity also. For example, for the natural media that can be used as a buffering agent, that function as a buffering agent, example is constip liquor. Constip liquor contain peptide, amino acid that will help to maintain the pH of the medium. Constip liquor contain peptides, amino acids that will all maintain the pH of the medium. Natural medium, it has a dual function that will act as a buffer also and it will also act as a nutrient also. For example, the corn strip liquor, if we are adding the corn strip liquor, it will also function as a nutrient material. It will function as a nutrient for the microorganisms, and will also function as a buffering. The second, that we also add the artificial buffering agent. For an artificial buffering agent, the example is phosphate. We can add phosphate to regulate the pH. Okay, we use the phosphate as a buffering agent, but the high concentration of phosphate interfered with the product of secondary metabolites. We can also use calcium carbonate, but the problem of using calcium carbonate is that if there is a below pH 7, then the pH decreases, the calcium carbonate will decompose. The balance use of carbon nitrogen ratio control the pH. If we balance the carbon nitrogen ratio in the fermentation media, that's help to control the pH. pH control extremely by the addition by adding ammonia, sodium hydroxide and sulfuric acid. pH may be controlled by the addition of this ammonia, sodium hydroxide and sulfuric acid. This is what all about the buffers that regulate the pH in the fermentation medium. Sometimes the natural medium has its own buffering capacity, or sometimes we add the artificial buffering agent such as phosphate, calcium carbonate, ammonia, sodium hydroxide and sulfuric acids. These all are the we add to regulate the pH of the medium. Now we will move to the precursor and inducers. Precursor, if we talk about the precursor, is not used for the growth, but it is used for the product production. Precursor, it is not used for the growth, but it is used for the product production, or we can say that is a product formation. It is used for the product production and it is used for the product formation, but it is not used for the growth. The things we have to remember about the precursor is that precursor that are added in the fermentation medium that regulate the product production and the product formation. Rather, it does not support the growth of microorganisms. It will not support the growth of microorganism. The well-studied example is penicillin production. In the penicillin productions, as we know that the structure of penicillin is somewhat like this. Okay, this much part is the thiazolidine ring. Okay, this much is the beta-lactam ring, and here is the acyl side chain, that is an R group. This much part is always constant. Thiazolidine ring and beta-lactam ring part, that is known as a 6-APA. That is a 6-amino penicillinic acid. This much part is always constant in penicillin structure. Always the R group vary. So, in the fermentation medium, if we add phenylacetic acid as a precursor, here it was the R group. Instead, in the R group, if we add phenylacetic acid as a precursor, then we obtain the penicillin G. Penicillin G will obtain. If we add the phenylacetic acid as a precursor in the fermentation medium, directly we add phenylacetic acid as a precursor in the fermentation medium, then we obtain the penicillin G. If we add the phenoxyacetic acid, then we obtain the penicillin V. It does not support this precursor will not support the growth of microorganisms. The things which we have to remember, that will not support the growth of microorganisms but help in the product production. If we add phenylacetic acid precursor, then we obtain penicillin G as a product. If we add phenoxyacetic acid, then we obtain penicillin V. Now we talk about the inducers. We know that natural mechanism of enzyme, that is enzymes are of two type: housekeeping enzyme and constitutive enzyme, or we can see that inducible enzyme. There are the two type of enzyme, that is housekeeping enzyme and inducible enzyme. Housekeeping enzyme, they are always present at some copies of one or two copies in the cells. Either the substrate are present or not, they are always present in one or two copies in the cells. Whereas inducible enzymes, they always get induced at a particular substrate. When we add the particular substrate, then and then only it will get induced. Inducers will regulate. Inducers will regulate the gene expression and allow the product formation. That will regulate the gene expression. Inducible enzymes are synthesized only in the response in the presence of inducers in the external environment. In the external environment, if there is a particular inducer, then and then only inducible enzymes are synthesized. For example, here we have taken, if the inducers molecules are such as starch and maltose are used as an inducers, then amylase enzyme will synthesize. The product will be of our amylase. If the inducers are maltose, then the product is the pullinose. If there is phenyl acetic acid as an inducers, then penicillin acylase enzymes that will are product. If various proteins are used as an inducer, then protease will synthesize as a product. If there is a cellulose as an inducer, then cellulase enzymes will be our product, will synthesize. If there is a pectin, then pectinase is an our product. These all are the inducers. Against the inducers, our product will synthesize. If there is a maltose, then pullinase will synthesize. If there is a various protein, then protease will synthesize. If there is a cellulose, then cellulase enzymes will synthesize. If there is a pectin, then pectinase enzymes will synthesize. This is what all about the precursors and inducers. Precursors that does not take part in the growth of the microorganism but support the product production. Whereas inducers, they are always synthesized when there is a inducible enzymes are always synthesized in the particular inducers are present. For example, if there is a pectin as an inducer, then pectinase enzymes will be synthesized. Now we will move to the inhibitors. The main function, the main work of the inhibitor is to increase the desirable product, used to increase the desirable product and decrease the undesirable product. The best studied example is the tetracycline. In case of tetracycline fermentation process, the undesirable chlorotetracycline formation was formed. During the tetracycline production, undesirable chlorotetracycline product formed. To prevent the formation of this chlorotetracycline, we add the chlorination inhibitors. What we add? Chlorination inhibitor, example that is bromide. In the tetracycline productions, undesirable product that is a chlorotetracycline was produced. To prevent the formation of this, we add the inhibitors such as bromide. Bromide is a chlorination inhibitors. Then we obtain the tetracycline. Like that way, the best studied example is the microbial production of glycerol. By modifying in the glycerol, by modifying the ethanol fermentation and by removing the acetaldehyde, inhibitors here we use, that is a sodium bisulfite. The bisulfite will react with acetaldehyde and that will form the acetaldehyde bisulfide. This sodium bisulfide will react with acetaldehyde and form sodium hydroxyethyl sulfide, or in the another word we can say that acetaldehyde bisulfide. So during glycolysis, dihydroxyacetone phosphate will act as an hydrogen acceptor and glycerol 3 phosphate will convert into the glycerol. The things which we have to remember about the inhibitors is that inhibitors are added in the fermentation medium to increase the yield of desirable product. Increase the yield of desirable product and reduce the yield of undesirable product. Here the examples we have taken about the glycerol and tetracycline. Here we use, these are the inhibitors, we add this type of inhibitors. Now we will move to the next step, that is antiforms. Antiforms, or we can say that antiforming agents we use in the fermentation. Forming due to the component of the medium. Forming is mostly due to the component of the medium that we are using. For example, if our medium containing corn strip liquor, peanut meal, soya bean meal, yeast extract, meat extract, peptone, etc. If our component is of this example that I have taken, if it is a proteinic in the medium, that ultimately leads to the problem of forming, that will create the form. As we know that the protein has a nature of that in the normal it when it combined with the water or any of those things, that will create a form. So ultimately the forming is due to the selection of the medium that we have selected. So while selecting the media, we have to take care of those things that does not create the forming problem. While if there is a problem of forming, we can go for, first we change the medium composition. Our main criteria is the first we go for the changing the composition of medium. We change some of the physical parameters such as pH, temperature, aeration, agitation. Due to the vigorous aeration and agitation, it also creates the form. Vigorous mixing, if we vigorously mix the component in the fermenter, then that also that time the forming may create. We can also use the best approach, that is the anti-forming agent. We use the anti-forming agent against the form. We can also use the form breakers. There are also form breakers and anti-forming agents. These are the standard approach to overcome the forming problem. To overcome with this forming problems, we use the anti-forming agent and form breaker. The characteristic of antiforms. Okay, whatever the anti-forming agents we are using, it has some characteristic. I think this much part is clear. Forming is normally due to the whatever the medium that we are using. If it is a proteinic medium, that create the forms, or due to the vigorous agitation and the aeration, that leads to the form formation. So to avoid the form, we go for anti-forming agent and we go for form breaker. And the first thing that we go for the modification, in that change we go for some change in the medium. Change the medium. So what are the characteristic of anti-forming agent? Whatever the anti-forming agent we use in the medium, in the fermentation medium, what are the characteristics that should be fulfilled? Should disperse readily and have fast action on existing form. It should have the fast action on the existing form. Whatever the form is been created, has been formed, it should has a fast action. Should be active at a low concentration. Should be long acting in preventing new form formation. should not be metabolized by the microorganisms. Form should not should be non-toxic to the microbes, human and animals. It should be non-toxic to the microbes, human and the animal. Should not cause any problem in downstreaming process. Form should not cause any problem in the downstream processing, means during the recovery process it does not cause any kind of hindrance or any kind of problem. Should not cause any handling hazards. During the handling it does not cause any kind of handling hazard. Whatever the foam that and antiforms we are using, it should be cheap. It should not affect on oxygen transfer, as we know that if there is a aerobic fermentation process, at that time we need to provide the oxygen. So at that time it should not affect the oxygen transfer. It should be heat sterilizable. While we are sterilizing our media, it should be heat sterilization. This is all about the fermentation media.